I'm going to start recording.
Okay.
All right, we're live.
Okay.
I
All right.
am I'm Professor Craig Story, or also known as Dr. Story at Gordon College, and some friends of mine, the Browns, and their friends down in Pennsylvania, have come up with a brilliant idea of doing a little question and answer today about basic biology, things that are related to the coronavirus that could help us understand what is a good response and how we should react in this time. And so, Pollyann, why don't you just quickly introduce the folks we have here today?
Okay. Pollyann, Dave Martin is here with us. He's a retired biology teacher of other subjects as well. My husband, Ken Brown, infectious disease specialist, retired. Peter Schreck is a retired professor of Palmer Seminary and a continuing family and marriage counselor, and his wife, Carol Schreck, also retired professor from Eastern Seminary and also currently still in counseling, marriage and family counseling. And I'm a retired educator. And in my earlier life, I was a registered nurse, but that's long forgotten.
Wow.
So
we have so many years of experience here gathered. It's wonderful, and yet none of us really have a lot of experience with even this technology. And as you can see, one of us has a computer that's not able to support Zoom, and yet we found a workaround. And so this is what we do. It's very exciting for me to be able to, and a privilege to be able to share with you guys and talk with you. And you came up with a list, Dave, I believe, came up with a list of say 11 or so questions. A lot of them are sort of basic biology questions, and I don't want to get too technical. I'd like to be able to provide information that helps people kind of appreciate what's going on, maybe as they may be suffering from this illness. To help to know what's going on can I think help even psychologically to understand what's happening. And so then at the end, we will leave it open for some of your practical questions, any questions you guys have. So let's see. So basic questions about the biology. So comparing different kinds of cells, bacteria, human body cells versus viruses. And that is a very good question. These are sort of various levels of organization within the world of biology, within the world of life science. The most complicated, in a way, are our cells, because they're much bigger. They have a metabolism. In other words, they transform energy. They have their own DNA information. A bacteria is a much smaller version of a cell. It's also still a cell with its own bacterial metabolism, taking in nutrients, processing, living, doing its thing. And bacteria also has a DNA genome. In other words, all the instructions for making itself and reproducing itself are contained in DNA. Everyone has an idea what DNA is. It's the information that encodes that what you are. Now, viruses are even more simple, and many people for a long time thought viruses aren't really alive. They're not alive. Nowadays, it's more of a semantic question of whether they're alive. And since people talk a lot about whether they can kill them with various disinfectants, if you can kill something, it's alive, QED, right? Logically. So for all practical purposes, you might as well consider them alive, but they are not cellular life. They are a viral form of organism. Well, they're not an organism. It's easy to slip. So organisms are defined as being cellular. Viruses are not cellular. They're subcellular. They basically are a packaging system for maintaining their genetic information. They use a cell to make themselves, to remake themselves. And they do this very efficiently and therefore spread very easily through the population. Viruses are, if you look at them, there are many kinds of viruses. Some of them have DNA genomes. Some of them have RNA genomes. RNA being another kind of genetic information that's very similar to DNA, but also very distinct from DNA. In order to replicate themselves, they get into cells and they are able to use the cellular machinery to make more copies of their genomes. They also use the cellular machinery to make the proteins, the structural parts that package that DNA or RNA into the virus that then buds out of the cell, and then it's more viruses. One cell can produce thousands of progeny viruses because they're so much smaller than a cell, and that's why they take over our bodies so quickly. Question 2 leads off from that, and that is entry points. Are there any serious questions with what I just said, or things that are really confusing to you before I move on?
No.
Yeah. So our cells then are these large, much larger parts of our bodies that are making up tissues, and our lungs, in this case, are the entry point for this particular coronavirus. You've seen pictures of the coronavirus having knobs sticking out of its surface. Those are made of protein. Those proteins bind to specific other proteins within our lungs, right? It doesn't bind to our skin per se, but our skin can then contact our face and somehow find its way to our mucous membranes and get into our lungs. When they hit a cell in our lungs, one cell gets infected, then you have 1,000, then you have 1,000 times 1,000, and very soon you have an overwhelming amount of viruses deep in your lungs, and apparently it starts deep in the lungs and moves its way up. This is why the early symptoms of coronavirus are unlike that of a typical cold, which is in the head and upper nasal passages. This one goes deeper in the lungs and then works its way up. And so the reason why certain cells are attacked by specific viruses has to do with that idea that the knobs on the surface of a virus bind to specific other proteins within the body and target those cells, those types of cells. Yeah. So structure number 3 has to do with the role that the subcellular parts have in manufacturing viruses. But let me pause there.
Yeah, I have a question. You said that the virus needs to get to the lungs because it binds there with some protein that it finds. Does it only attach to lung tissue?
As far as I think anyone really knows now, the major receptor is, I think it's an angiotensin converting enzyme. That's a fancy word for an enzyme that's a protein in your lungs.
Okay,
and it looks like men have maybe more of it than women, which may explain why men seem to do worse. A lot is preliminary. Now, with HIV, a different virus, human immunodeficiency virus, that binds to a protein on the surface of a certain kind of immune cell. And later on, after much time, it was discovered, well, there's other proteins it also uses to bind. And so there may be others that we just don't know about yet.
Okay, thank you.
Awesome.
Another question?
Yeah.
Can a cell be infected with 2 different viruses at the same time effectively? For example, flu plus SARS-2?
Oh, yeah. Yeah, no, that would be a sad state of affairs. Certainly that can happen. We know that happens by inference in the flu, because one of the mechanisms whereby seasonal flu can become more novel is to obtain genes from other species, ducks, pigs. So basically birds and humans and swine all have this, they can all be infected by the flu. And when a single cell gets multiple different species in the same cell, a new virus can bud out that has a mixture of bird and human flu. So it may have some aspect where the cell surface knobs are the bird variety, where we don't have antibodies ready to go against those. They're foreign enough to us, and we have a more severe flu season as a result. So yes, formally this can happen, although I think what tends to happen in a typical season in a typical town or city is some virus is going around. Lots of people get that. So all this worry and concern about testing, well, it's very, very important, especially in the case of coronavirus, because no one has experience with this. No one has any immunity. We need to test, and see where it is and try to isolate those people. But what was I saying? But you can presume after a while, if enough people are are being infected in an area, then you can, by the symptoms, assume with pretty reasonable accuracy that the person has that, whatever it is that's going around. You see what I'm saying? I don't know, Ken, you're a doctor, and some of you guys are medical doctors. Does that agree with your experience that certain things go around and then they?
Sure, and most of the time we don't ever know what they are. There are more than 200 cold viruses, and they're in several different groups. So
True.
Nobody has the time or the money or the interest to chase each one down.
Correct.
I think you're going to talk about vaccines a little bit, but there is a cow coronavirus.
Oh.
And there's
coronaviruses of all species. There's the one that this one happens to probably come from a bat. I mean, the sequence of the gene we've known for weeks now. It's the basis for making the vaccine. But so, yeah, nature. So all of these virus families, whether it be even HIV, came from two different times independently, actually, from species of monkey type, monkeys basically. And so, yeah, these virus families are found in all different kinds of animals. Chicken pox, monkey pox, all, camel pox, they're out there everywhere. And normally those other species don't harm us, but when they jump into humans and find a way to infect humans and become spread through the air or through surfaces, that's when you can have a potential problem. And what makes it worse is if people don't feel all that sick at first, and that makes it even worse because then they spread it more. So all these factors go into making it a more serious situation.
And Craig, one other question. How is it that this virus starts deep in the lungs? I mean, what's the deal with that as opposed to others that start up higher maybe?
I think it just simply has to do with the fact that the receptor is found more in the lung tissue itself rather than the upper airway. That may be part of it. This is something I heard an expert speak about. I don't have firsthand knowledge of that, but it sounded like they knew what they were talking about, that it seems to progress from the lung up. It definitely is consistent with the kind of symptoms that people get when you have a fever, that means your immune system is reacting and already fighting off the virus. And so for you to have a fever, it means you have a certain amount of virus happening in your body already. And it seems like fever comes on early on before you have any of those normal cold symptoms. So that's probably the kind of logic they're using to infer that there's stuff going on in the lungs before it gets up to your head.
And therefore you can share it more readily because you're not showing the signs as much.
Yeah, and also this cough is when you cough, little tiny droplets fly out of your mouth, and even energetic speaking can cause little spittle pieces to fly over. And it's kind of gross to think about, but these are the things that when you're sick are flying at you, and this is why people wear face shields, masks, and those masks are not 100% effective at reducing it. Just it minimizes and lowers the chance of catching it. Our healthcare workers are really on the front lines, and they are heroes right now. Really, they're putting themselves at great risk, but they have to really. Otherwise, society falls apart. But for them, here's to them. So,
can I jump in with a question?
Yes.
Okay. In other words, and just to piggyback on the why the virus is latching on to the lung tissues and the cells in the lung, would it be accurate to say then the specificity of the cells in the lung, which have particular jobs to do, just as the muscles in your legs have special jobs to do, each one of those cell membrane surfaces have a different texture and a different arrangement of the proteins on it that will then either reject a virus or accept a virus. And at this particular point, those lung cells are receptive
Yeah, that's exactly right. Yeah, so it happens to be that the lung cells, some cells in the lung, have enough of a concentration of those particular receptors that allow, more than likely what's happening is the virus has these knobs and it clusters those, it pulls the receptors together and sort of is able to really latch on there, and then that triggers the cell to pull that inside the cell. So there's a thing called receptor mediated endocytosis. So it's endocytosing, it's bringing it into the cell,
Right.
and that's the trick that the virus is doing to get in.
In Bio 101 is like active transport.
Yes.
As those molecules are pulled through by that receptor protein, then on the membrane surface to get into the cell.
Exactly.
Yeah.
I know with influenza, there's a after it gets in, there's even a mechanism for it to get out of the vesicle and release the contents into the cytoplasm, into the sort of the fluidy part of the cell, to escape that vesicle that went in and get out, and so then it can replicate. Now, the biology of the coronaviruses may not be as well understood as certainly as influenza or even HIV, but so many people are studying it right now, and one of the hopes that there is that we will come up with ways of interfering with its biological pathway. One obvious solution would be to make proteins that bind that interfere with the binding to this receptor, and I'm sure people are making those. Another thing is to make antibodies that block and bind to the surface of the coronavirus, and therefore also block its uptake into cells. So if you can interfere with that, that is one of the strategies that can be used for lowering the amount of infectivity and
Right. And I guess that's what the labs are doing right now.
Oh, they are. Lots and lots of labs are doing that. So
kind of a chemical.
yeah, they're doing it at a record-breaking speed, and the thing we really need to do is to keep the infection level down for a year and a half, or maybe quicker than that, if they can see some degree of efficacy. They do need to do safety trials. There have been cases, specifically with dengue, I know of, maybe others, but where the vaccine causes worse illness to occur in the vaccine recipients compared to not. Now that's a rare thing. My first thought was just inject people with this protein. I mean, what's the harm that can happen? Well, the harm that can happen is if it actually does cause people to have worse course of illness. Not likely, but it's not without precedent. So that's why it takes time before we're going to inject this vaccine into millions of people, to make sure that it really does have efficacy, but also safety. Safety is more of a concern than efficacy. Making an immune response isn't that difficult. Most things that are foreign, you inject them into someone in the right context, and they will make antibodies and so on. But making sure that it's also safe is important.
And
Now,
the current
virus, and once it gets into the cell, is replicating itself over and over and over again. And the viruses are just basically packets of really, I guess, in the coronavirus, just RNA. Or is there DNA involved in that?
Yeah, that's a good question. That's questions 4, 5, and 6 on your list. So
Right.
thank you for that. So the question is about the nature of the what happens to the virus when it enters the cell, and how more viruses are made. So yeah, so
before you go to that, I have one more question.
yes,
Greg.
Peter.
Are human lung cells pretty homogeneous, or like you said something about men get it more than women? Is that because a difference in the lung cells, or is it in the process of getting in and out, or what does that do?
Yeah, so this is all related, obviously. So yeah, so the exact difference in the biology between different humans is very interesting, because you all know that young children have much less disease.
Right.
They seem to catch it and be really good spreaders, but they don't get disease. And the thought of that has to do with this angiotensin converting enzyme. I wanted to actually spend a little more time researching that, but I haven't gotten to that yet. I just heard that on one of the doctors was speaking on the radio. I found NPR has a lot of good interviews with experts that has helped me a lot to understand this. But the idea being that children may have less of this amount of this protein that's expressed on the cells. It may increase with age. The reason older folks do more poorly generally is an effect of the lower activity of the immune system. That is maybe an independent factor compared to this receptor-related issue. So maybe men and women also have a difference. That was a speculation. I don't think that's been proven yet, but certainly in China, many more men died than women. I mean, many more. And they think that had a lot to do with the fact that more men smoke than women. And smoking is already a problem with going to cause your lungs to be less healthy. And it seems to be a very much a risk factor for this too.
Thanks.
So, yeah. So when viruses go in, their whole job is to manufacture their proteins, and proteins are these complex molecules that package things. They do all the work. They do all the enzyme work, most of it in the cell. And so they just need to make their own proteins using their own genetic information. And in this case, it's an RNA molecule that will specifically bind to these proteins, get packaged, and then be able to burst out of the cell. And so that process of bursting out of the cell is what leads perhaps to a lot of the illness, a lot of the symptoms. And that is you're breaking cells. Your cells are dying through this process. The cell doesn't like it. It's unhappy and bursts open, releasing its contents. And cellular contents, when they release in the body, lead to a process of inflammation. Your immune system has detectors for this, for when there is necrotic damage or killing of cells in a sort of sudden bursting kind of a fashion. And so this leads to your immune system being very activated. And if your lung is a fairly large organ, if this is happening throughout your lungs, all this material is being released, your immune system gets really revved up and makes you feel really horrible. You get fevers. Things like interferon get produced by your immune cells, and this causes you to have a lot of the symptoms. In fact, one of the things, an irony here, is that your own immune system's response may be the thing that actually kills you. If your lung is filling with fluid and that's part of your immune system responding. So this brings up another strategy to keep people alive, is to tampen down the overreactive immune response, blocking certain molecules of the immune system, maybe IL-6 for the really aficionados. So blocking certain immune responses to keep people alive long enough so that their immune system can produce its own antibodies and T cells and cells to be able to get past the hump where their immune system can then eradicate the virus. And so you have number 7, viruses. Number 6, how viruses make more viruses. Well, viruses don't make more viruses. The cell makes more viruses using the genetic information of the initial virus. So one single virus gets into the cell, and then it makes maybe 1,000 offspring viruses.
The cell does that.
And those are the factors, I guess, all of the bases, what they call the nitrogen bases, cytosine, guanine.
Yes, yeah. So DNA would be a double-stranded molecule, twisted ladder. RNA is basically like a half of a DNA, and it has a slightly different sugar, very, very similar, but the bases also are very, very similar. In fact, DNA and RNA can pair up with each other. If you're doing lab tricks, you can get that to happen. But they're very similar molecules, and normally in the cell, the nucleus of our cells has the DNA, but then that gets converted into RNA, which is then the instructions for manufacturing different parts of different proteins and things. So it also has structural functions and enzymatic functions on its own. The RNA does. It's really a world of new things that have been discovered in the last 20 years. RNA does a lot by itself, but the key message is that the DNA in our cells is basically the master recipes for making all these RNA, which is doing the important work. So it makes sense that a virus could have just an RNA genome, because really RNA is the important molecule. You really need to make the RNA to do
Getting
stuff.
the components of the other strand of the RNA actually then from the cell itself. They're actually feeding on that cell, so to speak.
Yeah. So there's plus stranded RNA virus, minus stranded RNA virus. The bottom line is that you can use that RNA information and make more of itself, and then package that and go on and make new viruses. Yeah.
Another question.
Yeah.
How fast? When you try to grow E. coli, you put one cell into a 5 milliliter broth, and overnight you get 10 to the 7th or a billion copies.
Oh,
How about viruses?
Oh, well, yeah, it's exponential growth. So the great illustration of exponential growth is, say you have a pond in your backyard and there's some kind of lily pad that grows every day. You have twice as many, and say, well, how does this go, right? So it's like on day one you have one lily pad, then you have two lily pads, and say the whole pond is full of lily pads on day 10. Like, when was it half full? Day 9, like the day before. Like, so it seems counterintuitive that you would go from half full to completely full when it's just one lily pad dividing every day. But that's the nature of exponential growth, is that at first you like barely notice these lily pads, and all of a sudden, hey, where did all these lily pads come from? And then by then the pond is already full. So yes, exponential numbers in the billions are nothing to a biologist. You know, these are tiny molecules, tiny viruses. If I spin down a million, 10 to the 6 cells, it's just a little tiny pellet in my tube, you know, a million cells. So we're talking exponential numbers of tens of millions of these little viruses making us feel sick. Yeah, when I was working on bee viruses this summer, there's one called deformed wing virus, DWV. And in fact, we were detecting this virus. We had our own little test kit that we discovered ourselves. In fact, it's not that hard to make your own test kits, ladies and gentlemen, but you have to have a few things. You have to have some access to different enzymes, and you have to order some DNA and make some primers. You have to have some knowledge, but it's not something that is an impossible thing to do. It's shocking, and literally stunningly shocking, that we have taken so long to put out test kits. Part of it is just the lack of directives, lack of organization. The science is there, and look what they did in Korea. You know,
yeah.
it's just shocking that we haven't done that. What was I going to say about that? Yeah, so in the summer we had we were detecting viruses in beehives, and we were detecting, we were getting like 10 to the 10th virus particles per, very small volume. Just like the amounts of viruses that you're talking about are very, very large numbers that will make something sick. You take a bee, crush it up, and you have, 10 to the 10th virus particles per mil or something like that. Very, very huge numbers of viruses. Yep.
Ken, were you asking how quickly this happens?
Yeah, I was thinking about I guess the phenomenon of doubling the growth probably is best illustrated for viruses when one day you have a rash and the day before you didn't have a rash.
Sure.
Which implies that all over the skin, for example, in chickenpox, for example, it comes in crops. But nevertheless, one day it's there, and the next day one day it isn't there, and the next day it is there.
That's right. And
Sudden multiplication.
if you think about viruses that we have in the human population, chickenpox, or what's another virus? Well, even the influenza, human influenza. Normally, it doesn't kill most people, and it can be controlled by the immune system in a window of time before it could kill you. Now, if we didn't have an immune system, all these viruses would just kill us. We have no chance. But they're sort of in a balance with us. There's a balance between the virus reproducing itself at a certain rate But if it's too high, it will kill the host, and then it won't be able to spread. So the virus, from the virus's point of view, which it doesn't have a brain, it doesn't have intelligence, but it's to the advantage of the virus to not be too aggressive and too good at replicating. And so there's a balance over eons of time that develops. These viruses develop a relationship with their host species so that they sort of maintain a little bit of an agreement. All right, I won't make you that sick, but you're going to agree to, you know, go ahead, fight me off, but I'm going to spread to a few other people, and I'm going to stay alive. I'm going to keep, in a virally way, stay alive in the population. And so so this is why all pandemics that I know of come from another species where the virus doesn't make the bats all that very sick, right? But then it jumps into humans, and it makes us sick because it's not a human virus. We don't have that experience. And so part of it is the lack of our immune system's experience. Part of it also could be that it just actually is more aggressive in different host species.
You've mentioned it several times here in your discourse, your immune system. Can you address that just for a minute or two?
Yeah, that's question number 10 and 11. That's our final two questions. So you're asked in how do vaccines work? And also, once infected, can you be infected again? Those are really important questions. So a vaccine typically works by mimicking an infection. So in a typical infection, we build an immune response. And our immune response is a bunch of cells that recognize a foreign invader and create defenses to fight it off. This includes antibodies, which most people are familiar with, proteins that are specific to that illness. But there's other things that are made as well, including cells that will kill and get rid of infected cells. They can detect that a cell is infected and kill that cell and thereby kill that virus. So lots of different kinds of immune responses occur. And the key ones that help you with viruses tend to have a memory to them. They create cells that respond specifically to that virus, and then they persist in your body for long periods of time. We're talking years. They can stay around for years, and they keep when that virus comes along again, or one similar to it, you have an advantage because that cell can be more easily reactivated and very quickly respond, so that it doesn't take you 2 weeks to fight it off, but maybe in 4 days you're already fighting it off and you just get a little stuffy nose and that's it. So vaccines work by mimicking an infection typically, and you as a result produce cells that stick around for a long term, and they also produce antibodies that give you that the ability to block the virus entry, as well as other cells which can directly kill infected cells in your body. Now, the immune system fights off viruses, fights off bacteria, fungi. Some of the defenses are different for those different kinds of players. And it even fights off cancer. Precancerous, small little clusters of abnormal cells are being taken care of all the time by your immune system. Lots of evidence that is a normal function of the immune system. So once infected and you survive the symptoms and effects, it's unlikely that you would be infected by the same virus again, at least for a year or two. Depends. Some viruses are good at blocking our immune defenses, and they can cause perhaps some kind of reinfection at an earlier time. But for all we know, it's very likely that you would be protected, which actually is good for healthcare workers. If they know they were infected and they recover, then they are the ones that are like, they should get a bonus, a pay raise, and they don't have to worry about getting it again or spreading it to people because they will have controlled the infection. Things that need to be looked at are like, how long does a person continue to release virus after they're infected? All that stuff can be looked at by these very sensitive DNA amplification tests. They take the RNA, convert it into DNA, and then amplify the DNA. That's how a lot of these tests are working right now. So was that clear enough, Pollyann?
Oh, Dave, are you Did you have your question all answered?
Well, go ahead, Pauline.
I just was wondering about the anti-vaxxers and how that might feed into this eventually. If we don't know how long, you know,
Yeah, there's a lot of people that fear vaccination, and you know, a lot of times it's a result of misinformation, or a lack of kind of appreciation of the danger of the disease that's out there. Often they'll say, well, I'd rather just have my child get the illness. Great, but what about them giving it to someone else who may not be able to handle it or survive it? So I think an objective look at public health and history should lead anyone to realize the importance of vaccines as a public health measure. And you know, I said this a couple weeks ago to somebody. I said maybe this experience will just shake up that anti-vax movement, and if they can see how effective a vaccine can be at helping us to fight off this unseen enemy. Polio, right? I mean, you guys know the polio outbreak. What year would that have been? The 40s, 50s?
1959. Well, the outbreak. I thought you meant about the vaccine.
Yeah,
it was the 40s. It was the 40s and
Yeah, it was
early
while we were
50s.
growing up.
Yeah.
Dog
yeah.
days and polio. You
Yeah.
couldn't go swimming in August.
Yep, people were drawing the curtains and hunkering down in the home, and you know, the anti-vaxxers are like, we don't want to give our kid the polio. You know, like it must seem crazy to you guys because you saw the effectiveness of the polio vaccination, and there's still countries, maybe 5 or 6 countries in the world that still have polio, and it's because their governments and the people in the countries don't allow public health workers to go in there and do their work. It's a political reason why we haven't eradicated polio, which is really unfortunate. So I'm hoping that anti-vaxxing movement will subside.
Yeah.
On Facebook, I see people that are sticking to the story like, well, let's just see how dangerous this really is. I mean, well, we don't have enough evidence. Honestly, the evidence is there. It's right there. You know, look at China. Look at these other countries. Look at Italy and Spain right now.
Mm-hmm.
They're about 8 days ahead of us or so. So I think, you know, luckily we finally have gotten people to pay attention. Look, don't meet in groups of 2 with people. You know, people say, well, now it says a group of 10. Oh, great. I can have my group of 12 because it's close enough to 10. I mean, the point is minimizing any kind of contact with people that you don't know. They might have gone to the grocery store, got infected 3 days ago. Charts and graphs. I saw one chart and graph that showed, I think it showed the amount of symptoms and the amount of days after infection. And about 50% of people had no symptoms 5 days after infection, which means they probably are giving off virus and not showing symptoms. So that's very dangerous.
So
I really liked your analogy that you used, that the vaccines are imposters of the actual infection, of the actual virus. And I guess really over time, the cells reproduce themselves, and they don't have those blockings with it. So will that over a period of time the vaccine wear off? Then was a whole new cells being produced there.
Yeah, so that's a great question. The question is, in case the sound doesn't come through, the question is, vaccines are in a way like an imposter, a foreign agent that mimics the infection, but doesn't make us sick. So the question is, how long will that last? What is going on in terms of these cells that are being produced? What happens is you make a specific response. These specific cells to that particular vaccine, they expand greatly, and then most of them die off when they're no longer needed. But a certain subset of them stay in the body and stay alive for years, and perhaps they do divide at a slow rate, but they remain and like in the bone marrow, they remain sequestered away. They circulate around the body and sort of retain that memory. Part of so again, based on not all vaccines are equally effective at generating the memory cells. In such cases, say a good example would be shingles vaccine, or also known as chickenpox, same virus, VZV, varicella zoster virus. This one needs a couple of boosters, even for your primary vaccine for kids. It's just not very effective at generating the kind of a robust memory response that you need. Polio vaccine, on the other hand, I think one dose of the oral polio does a really good job of giving you like lifelong immunity. So different vaccines are differently effective, and that's why another reason why we need to really do good testing on a vaccine. We want to use the most effective dose, the most effective dosing. Well, dosing, right? If you give too much of it, it might actually cause less of a response, and it may make it more costly to produce the vaccine. Not that that's the primary thing, but you may not be able to make as many doses. If you can use a lower dose and get a stronger response, that would be a win for everyone. So these are the kind of things that need to be done in the testing process, which takes on the order of a year. So let's all hope that we can find some solace and joy in this time, do more writing. You all talked before we started recording about how right now it's hard. It's hard to set down and do your work because of all the excitement, and it's a novelty thing. And I was encouraged that I thought people would be encouraged to learn that you more experienced folks are having just as much trouble as the college students, perhaps, at settling down and doing their homework and doing the things they want to do. But I think we'll adapt. It may just take a week or two, hopefully not that long, to adapt to this new way that we need to put up with for a little while.
How much encouragement do you think you should take from the fact that there are effective coronavirus vaccines for cows, for example?
Oh, I have no doubt. I have little doubt that we will have a good vaccine, and that it's eminently vaccinable, druggable. Is that a new word I invented? I don't know. But for example, HIV, we don't really have a good vaccine for that. This is a virus very different than HIV. For various biological reasons, it's much more likely that we can generate a vaccine that will work for this coronavirus. HIV, for example, infects the very immune cells of your immune system. And so therefore it's all right away harder to vaccinate against that. So I think there's a lot of good hope that this is going to come about. I didn't know about that cow coronavirus vaccine. Is that right? I know coronavirus causes feline disease as well as dog distemper.
Camels.
Yeah, it's widely found, and it's just unfortunate that we have been given this virus from the wild, from the bush, and it's sort of inevitable that this will happen. I think we may have encouraged it by certain practices, but we do. All cultures interact with wild animals to some extent. And so I think we need to re-examine some of that, but there's no point in, we have this problem. Let's just deal with it and fix it and help learn lessons from it. But yeah, good. I forgot the original question. Oh, will we find one? So what is this cow virus that you're talking about, Ken?
I don't know its name, but I know the vaccine is accessible. There's actually it's a GI disease, causes diarrhea. And one of the vaccines you give intranasally, which is unusual, and the other one you give by mouth. But it is reasonably effective. And I guess the demands for some animals aren't as great as they are for humans. For example, there's a mastitis vaccine that is good, but it isn't very good. And people still use it because it means a difference in making a living. So I think Peter Schreck might have a final question.
I have two questions at least. Can I ask them?
Sure. We're making more than a 10-minute podcast, but that's all right. It's interesting.
A question: the damaged lung tissue. will it repair itself back to a really well-functioning lung, or will these people tend to have compromised lungs?
Yeah, I heard an expert speak on that, and I guess it does depend on the severity, and there is a limited capacity of lung tissue to regenerate itself.
Mhm.
Read about that also with respect to the vaping trends that are happening. Some of these kids that have burned out their lungs are going to have chronic lung problems for the rest of their life. Now, that's not to say we may discover some kind of regenerative medicine that could help in the future, but right now, I think lungs don't fully regenerate really well. So, yeah, people might end up having compromised lung function.
Okay. And the other question I have is, I've not heard anyone talk about pets
I have.
in terms of being carriers and
Yeah.
distributors. Is that likely? I mean, do you?
I've heard of that. I heard a discussion of that this very morning, and I think it was Cy Montgomery, who's a writer. She's a kind of a science writer. You should read a book on octopuses, by the way. It's really good. Anyway, she was citing someone. It's just third-hand information, but I believe there were a couple of dogs in China where they tested them, and they were the dog pets of the pets, I should say, of someone who had coronavirus, and they had a mild positive. So, of course, this came from other mammals, right?
Yeah.
Bats to something to humans, and camels were involved in the MERS one, right? And so, of course, it's very possible that any mammal, even a household pet, could catch this very coronavirus. Now, but the evidence so far is that it's not a big thing. It's not a major thing. Now,
I was thinking so much catch it as much as carry it. I mean,
on the fur, yes. Yeah, on the fur,
Oh.
they lick you and they go around. So I can tell you a true story that happened the other day to me. I mean, I think the virus formally can persist on any surface. Now, fur is a very three-dimensional surface. It's not as easy for it to maybe transfer through fur. Say you're trying to make a print of say, an ink print of something. You would use a solid plastic thing and you would stick it in the ink and then you would print it and you get a nice. If you use fur, you're not going to get a very good transfer. So it's analogous to that. I think, though, if you if a person say I have coronavirus and I don't know it yet, that could have been me last Saturday. I'm every day that goes by, I'm more confident I don't have it. But I was in Florida last week with students. Was it last week? When was it? It seems like
2 weeks ago, yeah.
And I could have picked it up at a rest stop, and I could be on day 5 and giving off viruses. So my wife and I went to Halibut Point, which is a beautiful spot in Rockport, thinking we just get away from people, walk in nature, and see the beautiful world. And wouldn't you know, we get there, and it's just mobs of people, crowds of people, all spaced apart from each other in little groups, which was good. And yet it's a big enough area. I didn't feel unsafe there until something happened with a dog. An elderly couple walking along, and they had an elderly dog. And Kelly and I were sitting eating our crackers on this rock. And you got to picture this place is an old granite quarry, and there's a huge pool full of water now, but then there's these little hummocks of granite with scrub brush growing around them, and they kind of fall off steeply. And I'm sitting there with Kelly. We tried to pick one away from the trails. This dog comes tottering up, really happy looking, friendly little old dog, gray-haired dog. And I lifted my foot. Now, I didn't kick the dog. I just lifted my foot to block the dog because I didn't want the dog to come over and lick me or whatever, because I thought, man, maybe he'll give it what I have to these people, and maybe they've got it and they'll give it to, you just, so the dog was potentially a vector. And wouldn't you know, the dog, being a little elderly, just fell off the edge of the rock into the prickle bushes a little bit. And I felt bad, but I was like, hey, I don't want to touch your dog because I don't want to give you this virus. I explained myself, but it was kind of a silly situation that leave your dog on a leash, guys. You don't want little kids coming up petting your dog and then you catch it from them. Yeah. So public health concerns, all these things, like when you push the ATM buttons,
Yeah.
How many other people have been pushing that. I would recommend the podcasts from it's called The Daily from the New York Times. they have interviewed some of the top people. And starting last Friday, it was Friday the 13th, easy to remember. They had one that was just stunning about what's going on in Italy. And previous to that, they had living, or was that the one? Maybe that was the one, living with coronavirus. And they interviewed people that talk about what they do to minimize their chance of catching it. Now, well, the experts say most people are going to get it. That assumes that we don't have a vaccine. That assumes a lot of things, but we want to get it later on. We want to get it later on when the hospitals are not overwhelmed, like they are in some countries in Europe right now. So we want to shift the peak of the curve and squat it down so that it all occurs at a much later time.
So, Craig, are you saying, excuse me, that there will be no vaccine up before a year?
Yes, that's what I'm saying, but I'm basing that on the word of Tony. I didn't know he was called Tony. I always call him Anthony Fauci from NIH.
Okay.
And he said that, and that makes sense to me.
And what we're hearing from other sources, especially, is, well, we could maybe use this medication or that that's just bogus, right?
Well, yeah. So everyone's talking about these malaria drugs and these other drugs. The chance of any particular drug working is very low, but of course, everything is being tested. Everything's being looked at, and it's being done on a massive scale. As soon as something shows efficacy and shows some effect in a controlled study, it will be well publicized, and you got to believe that that person will be a hero. So there's a lot of various inducements for people to do this kind of work, and I think that particular study is certainly not an ironclad study, and we just have to wait and see. So I would strongly suggest against taking those drugs because they have side effects after all, and they're not proven. So certainly any homeopathic kind of remedies, things like gargling, drinking hot water, I've heard all these. They are like you said, bogus. They are highly unlikely to have any effect because the mechanism of how they would work doesn't make sense. And yeah, those kind of things are unfortunate that they're going around and people tend to believe them. But yeah, not yet. But as soon as something's available, we'll hear about it.
Okay, one other question. If the entry point is through the lungs, why are we not then, when we're out and about, wearing masks?
Well, because the very finest particles that are floating around, if it does aerosolize, so with flu, with influenza, the reason it seems to be more contagious in winter has to do with the dryness of the air and the fact that they can actually remain viable while in a form that floats in the air. If we had enough masks, I don't think it would be a bad thing to wear one. There are some advantages, but we don't have enough masks. Hospitals are running out of them. They're reusing them. So it's very important that people don't go and buy masks for themselves. They need to be in the hospitals.
I see. We have masks, but I've never even.
Yeah. I mean, the small amount of masks that different people have probably isn't enough to help the hospitals right now. But certainly what they can do is they can help. If you are talking to someone, it'll catch the little spittle droplets, things like that. That can help. It can help keep you from picking your nose or touching your mouth or chewing your nails, stuff that can just make it worse, right? It can help in certain ways, but it's not 100%. It's not effective at like blocking the finest air particles from getting in. It's not like it's 100% effective. But I think the main reason that people say not to use masks is public health reasons. They just started at Brigham and Women's and the Partners Healthcare System in Boston that all workers in the hospital are wearing masks during the time they're there. They're all wearing them now. That was a new policy change that just happened today. So obviously it's helpful for everyone to wear masks, but we don't have enough masks.
Yeah.
Yeah.
What about immune system boosters?
Yeah, you don't want to boost your immune system, ladies and gentlemen, because that is just a bad idea. Now, would you want to boost your digestive system? You know, that's diarrhea, right? You don't want that. What you want is just the right amount of immune system, the normal amount. You don't want to have a weakened immune system. How do we have a weakened immune system? Not enough sleep, too much stress. Those two things alone, if you can minimize those, you're in effect not boosting your immune system, but you're maintaining your immune system to the normal levels. A boosted immune system leads to autoimmunity, things where your immune system starts to attack your own body, arthritis. That's when the immune system is overactive. So I would suggest maintaining a normal immune function, but not trying to boost your immune system. So it could be when people say boost your immune system, what they really mean is maintain your healthy immune system. So for that, you do all the good things like you should do: exercise, eat a lot of vegetables, get enough good night's sleep. And all those things will only help you in your fight against the coronavirus or any other illness for that matter.
Great, thank you.
Yeah.
To end on.
Was that a good ending?
Oh,
Yeah, that's terrific.
All right.
so helpful.
Thank you,
Thank you
Craig.
so much, Craig.
Well, we can do this again in a few days, and I hope you all are staying healthy, and also in your community that people do take it seriously. And even groups of 2 or 3, they say family units, husband, wife, and so on. That's almost can't avoid being together. But if one is exposed in some way, then someone's got to move to the garage. In fact, doctors are doing that. They're like putting a separate set of clothes in the garage. They come home from the hospital, change out of their clothes, take a shower, all these steps to protect their children and stuff. So yeah, it's a real threat, and it seems crazy. It seems like, oh, I'm just going overboard, but really you really can't in this environment. So all right, you guys, great to meet you all.
Very much.
Thanks, Craig.
All right, I'll make this into a video.
Thank you.
Okay.
All right.
Are you okay with this going out into the world generally?
Yeah,
All right.
it's not a virus.
Maybe we'll go viral. I hope they change that term. Maybe it'll spread widely. Okay.
Thanks, and you stay well too, you hear?
Thank you. Thank you very much.
Yes.
All right. God bless. Bye now.
Bye-bye.
Bye-bye. Say hello to Kelly.
Will do.
Okay.
Okay.